Yeah. So in the previous section, we were talking about the different components of SASA, that is FOSA, PISA, PISA, and then we went on to talk about WPSA, that is weakly polar component of the SASA, that is halogen, the phosphorus and sulfur atoms that are present, they will be responsible for the weakly polar component of SASA. Okay, so this value should be in between 0 and 175. Then volume is another physicochemical property which talks about the total solvent accessible volume in cubic Armstrong using a probe of 1.4 degree Armstrong radius. So this value should be in between 500 and 2000. And donor hydrogen bonds, donor HB. So this estimates the number of hydrogen bonds that would be donated by the solute to water molecules in an aqueous solution, यानी कि by the drug to water molecules in an aqueous solution. So values are taken as averages over a large number of configurations. So this value should be in between 0 and 6. Okay, in Lipinski's rule of 5, we said this value should be in between 0 and 5, right? But here we are stating a new value 0 to 6. Then acceptor HB. So this is the estimated number of hydrogen bond that would be accepted by the drug from the water molecules in an aqueous solution. So values can are averages which are taken over a large number of configurations, and these values should be in between 2 and 20. In Lipinski's rule of 5, we said this value should be not more than 10. But now we are saying this value is in between 2 and 20 for a large number of molecules. Okay. Then this is square of the dipole moment divided by the molecular volume. Now this is a key term in the in one of the equations that is related for free energy of solvation. Okay, so this value should be in between 0 and 0.13. So we'll not get into the details of this Kirkwood Onsager equation and other stuff. Okay, then another value is another physicochemical property is this, which talks about the cohesive interactions in solids. Okay, and this should be in between 0 and 0.05. So in Ceutics, we study something of sort of tap density, bulk density, right? So those all parameters talk about cohesive interaction, right? That angle of repose, for example, they talk about cohesive interactions between solids. So this is another term which in medicinal and pharmaceutical chemistry talks about the cohesive interaction in solids. So this value should be 0 to 0.05. Then globe is nothing but a globularity descriptor. यानी कि even if we say that a molecule is linear, it tends to be globular, it tends to roll up slightly, right? When it enters into a aqueous environment in our physiological conditions, right? So that is nothing but globularity. So where R is the radius of the sphere with a volume equal to the molecular volume. So molecular volume जब consider करेंगे, तो if it has, it will turn into a globular structure, and उसका radius would be measured. So if the globularity is 1, it is a spherical molecule. And ideally for molecule, it should be in between 0.75 to 0.95. So more globular the molecule, more better are the chances that you know, it will be bioavailable. Okay, then we have QP. This is again a parameter for predicted polarizability in cubic Armstrongs. Then we have QP log PC. Now this is hexadecane gas partition coefficient. Initially we talked about octane water. Now we are talking of hexadecane gas partition coefficient. Then we have QP log POC, which is predicted octanol gas partition coefficient. So previously we had octanol water. Now we have octanol gas partition coefficient. QP log PW. So this is predicted water gas partition coefficient. QP log PO by W. This is predicted octanol water partition coefficient. QP log S is predicted aqueous solubility. So why are we talking about gas partition coefficients in between? Because we know some drugs are taken via via lungs, right? They're taken in the form of aerosols. So in that case, like for example, we have the general anesthetics, right? So general anesthetics are taken directly via inhalation. So in that case, we need to understand their gas partition coefficient, and that is the reason why gas partition coefficients are also studied. Log S is solubility in moles per decimeter cube. Okay, this is the concentration of the solute in saturated solution that is in equilibrium with the crystalline solid, and this value should be in between minus 6.5 to 0.5 plus 0.5. So minus 6.5 means it is heavily water soluble, whereas plus 0.5 is it means it has good amount of water solubility. Anything that is higher than water 0.5 would be, really bad as far as their water solubility is concerned. Then we have QP log S CI. So this is confirmation independent predicted aqueous solubility. Then we have log S, again the same thing that has been repeated, okay? And QP log HERG. Now this is predicted IC50 value for blockage of HERG K plus channels. Now what are HERG K plus channels? These are these are potassium channels which are present on the heart tissue, okay? So if the value of this value, predicted value for any drug, newly designed drug, comes to be below minus 5, so then that means it has very high affinity for the HARG potassium channels which are present on the heart. And that would mean that it will have heart related side effects. Okay, that would mean that this molecule has heart related side effects. So heart attacks and other, cardiac related uh, toxic effects or side effects would be seen for that particular molecule. Hence, any molecule that is designed should have a value of QP log HERG which is above above minus 5. Okay, then we have QPP CACO, then we have QP log BB, QPP MDCK. So we have studied about this, and here the values are also mentioned. Okay, so CACO 2 value should be greater than 500. QP log BB should be in between minus 3 to plus 1.2. QPP MDCK, this value should be minus 8 to minus 1. Okay, so then we have another, this should be greater than 300, and QP log P, this is another parameter. So that value should be minus 8 to minus 1. Okay, so that indicates skin permeability or transdermal route of administration. But QPP MDCK, the higher the value, the better would be the absorption across the intestinal barrier. Ideally, it should be above 500. QPP MDCK. Okay, QP log KP, this should be minus 8 to minus 1. Then we have ionization potential and its value should be minus 0.92 plus 1.7. Then metabolic, the number of metabolic reactions, it should be between 1 and 8. QP log KHSA is the binding to human serum albumin. It should be in between minus 1.5 to plus 1.5. Human oral absorption should be 1 or 2 or 3 for low or medium or high values respectively. Percent oral absorption that we have already talked about, right? Above 80% is good absorption. 20 to 80 is okay, but less than 20 is bad absorption. Okay, then yeah, so that is what we have written over here. Greater than 80 is high, less than 25 is poor. Then we have solvent accessible fluorine, SA fluorine, that is solvent accessible surface area for fluorine atoms for those molecules which have fluorine in them. Solvent accessible amide for those which have amide oxygens. Polar surface area is another important parameter. When we talk of the van der Waals surface area for polar nitrogens and oxygen atoms, this value should be in between 7 to 200. So if it is less than 40, that means the molecule will be absorbed into the CNS. It will cross the blood-brain barrier. If it is more than 40 up to 200, that means it will be orally bioavailable. So that is the importance of polar surface area, this particular parameter. This is very important. The number of nitrogen and oxygen atoms, it should be in between 2 and 15. Okay. The rule of 5, Lipinski's rule of 5, we already know, will not get into the details again. Rule of 3. This is another important. This is called as Jorgensen's rule of 3. Okay. Then we have number of primary metabolites, number of ring atoms, number of in-56, that is number of 5 or 6 membered rings. We have values for all of these. Non-conformer, non-H atoms. GM is the transdermal transport rate. Okay, so these are all the different physicochemical properties that are available. Okay, and we'll have our assignment in the upcoming session. Okay, I can see that almost everyone is left.